Hello, everybody. Welcome to Unexpected Biotechnology. I'm very pleased to welcome you here today. My name is Stephen Chambers. I'm Managing Director of Indie Bio here in New York and also a partner at SOSV. The session we have today is Manufacturing in the New Bioeconomy. I'm very pleased to welcome two guests that we've invited along today, Stuart Wilkinson, who's co-founder and technology director at BioBrew, and also Will Canine, who's co-founder at OpenTrons. Stuart has extensive knowledge of deploying back technology, particularly at scale and manufacturing. So I brought him along today to talk about some of the issues that we see with scale up and some of the problems that I encounter on a daily basis. Hello, Stuart. Good to see you.
Hello, it's a pleasure to be here.
Do you want to tell us a bit about BioBrew, Stuart?
Absolutely. So BioBrew is what I would say is a technology platform within ZX Ventures, and it's really exploring the biotechnology domain, what we call the beyond beer space. It's really focusing on food grade precision fermentation and downstream processing, with really an aspiration to unlock bioproducts and industrial food biotechnology production at large commercial scale.
So I'm using you as my myth buster, okay? So I'm going to be asking you a lot of questions. So one of the things I see with a lot of the startup companies that we see coming through the accelerator is that they have a particular host that they're working with. It's obviously they've got a certain amount of familiarity with the host. Can you just tell me a bit about the issues that you have with scaling up different hosts and what you should think about in the choice of hosts?
Absolutely. So it's a really critically key, important point, I would say, appropriate host selection. So there's much debate and a lot of searching for what I would say is the holy grail, looking for a universal best host. But unfortunately, there actually isn't a clear winner. It's very much case by case, depending on the specific molecule. With regards to specific selections of hosts, so things like the question of, yeast versus bacteria versus filamentous fungi or even mammalian cells. All of them have got strengths and weaknesses. What I would say is, look, things like bacteria, there's some strong points there. They grow very fast. It means that you've got fast fermentation times and it allows fast strain engineering cycles. But there's some key things to think about, like the likes of the risk of phage infection is a big fear of mine, especially at large scale. Things like how to deal with a decontamination sort of system if that situation does arise. And the need for backup strains is one of the key things for bacteria. I think jumping to sort of some of the other strain options, the host selection options, if we think about things like filamentous fungi, strains like Aspergillus, trichoderma, for instance, slow growth is one of the things to consider here, really. And slow growth means slower fermentation times. Slower fermentation times mean higher contamination risks. So it's really one of the key things to think about in a development program even, because slower growth means slower strain development. But there's also a handful of other issues with things like fungi, sporulation and how you would control that. There's some CMOs, contract manufacturing organizations we know, that are not so comfortable with filamentous fungi. So there's already, I would say, a bottleneck, a lack of scaled capacity availability. So using certain things like certain filamentous fungi strains can actually sort of further restrict the options that you have with regard to scale capacity. One of the other things to think about as well is expression levels. So the titers, let's say, of a protein or a product that you want to try and produce. It usually takes some experimental trial and error to find out, what the base level expression is. And a lot of the kind of reported high expression titers of certain systems, filamentous fungi being a good example here, they can be quite misleading. So I think in the case of certain things like homologous, native enzymes, things like fungi are very, very potent sort of expression systems and secretors. But I think when we start to look at things like heterologous, more complex mammalian proteins, I think it's a different story and it's much more challenging. So not falling into that trap of sort of assuming that one strain is better than the other.
What about the question that you always get Saccharomyces or Pichia? Have you any thoughts on that question?
Great question. I would say, look, both of them are actually almost equally as capable in the right circumstances. A lot depends on if it's intracellular, where it accumulates within the cell, or if the product has to be sort of extracellular, secreted. There are some proteins and some biomolecules where one or the other may be more sort of amenable to having higher secretion. But again, it's really got to be tested. And to your point, sort of not being, I guess, locked into a particular host organism just because of familiarity with it or more handling experience. You've really got to evaluate sort of all of your options, and likewise, even things like, mammalian cells on paper seem a really, really sort of really nice option, especially for complex mammalian proteins, things like dairy proteins, for instance. But there's a lot of challenges with mammalian cells. They're very sensitive to things like shear stress. There's a much bigger challenge of sort of avoiding contamination. And then certain other things are sort of like, one thing to bear in mind is really sort of what scale are you aspiring to produce at? Because things like chronological aging, so how many cell divisions, a particular strain or a host can actually go through before they start to lose that phenotype, the traits that you're looking for. And some of them are actually more limited than others. So if your intention is to go to what we would call in BioBrew large scale, which is sort of 500 meter cube to 1,000 meter cube, so 500,000 liters to 1,000,000 liters, that needs to go through quite a lot of cell divisions just to propagate for the seed train, to get enough biomass to be able to inoculate those. So if you're capped at a lower number of cell divisions, you may never actually get to the scale that you are, let's say, hoping to get to for the economy of scale approach.
So Stuart, when you go to those scales, how many steps do you have? Like, like how many, the pre-incubation fermentation and like you're not just putting a 1 liter flask into,
Absolutely, yeah.
Like, so what's the steps that you have?
So again, it's very, very case by case. So it depends to some degree on what your strategy is in terms of how you've developed the process. So typically there are levers to pull in terms of, you could say, right, I'm gonna inoculate my big fermenter with a high concentration of biomass. That gives you, let's say, a higher probability of success and a lower probability of contamination. The more established a colony that you inoculate into your big fermenter, the less chance there are for things like unwanted bacterial strains to get a grip of that. But with that, you need to go through an increased number of steps. So to grow that initial biomass, you've got more steps needed beforehand. So a lot depends on your overall techno-economics. But just to give you kind of like a rule of thumb, I'll use yeast as a sort of a case study there. What we typically see is anywhere from a sort of a 10x down scaled step. So if you're looking at something like a 500,000 liters, you might have a pre-seed scale, which is maybe 50,000 liters. And then below that, you could have a 5,000 liters. And beyond that point, a lot depends on sort of what capabilities. But it's possibly anywhere from 3 to 5 different growth steps in sort of a sequence, scaling up so that you get that biomass and that sort of big enough amount of biomass to be able to inoculate.
Another question that comes up with companies, obviously, we're working with early stage companies. Like a lot of the focus of what we do in the Accelerator is basically moving the technology out of the lab into kind of the factory or into the process side of things. And so we see a lot of companies come in with what they would describe as models. Oh, this is and they've built the model either in microtiter plates or deep well blocks, and they have a model of its production, of its expression, and have you. And they kind of think that can just be scaled up. Can you like as I've been telling you before, like in my own background, there was always two different schools. It was kind of like the guys in the lab that used to sort of start stuff off, and then there was like all the engineers who played on the fermenters, and they never did meet, kind of. So, like, can you just talk me through the process of what scale up looks like and how that works?
Absolutely. I think the first thing to sort of start with is, in terms of a sort of myth buster, is that upscaling, scale up is much more complicated than most people appreciate. You mentioned sort of microtiter plates and models, fantastic systems, great for screening microtiter plates, high throughput systems are fantastic, but really not representative of what I would call industrial scale. So again, to use kind of like the industrial food biotech sort of yeast systems at sort of 100,000 liters to 500,000 liters, those microtiter plates have, I'll be honest and say, very little relevance in terms of the process sort of dynamics to mimic those kinds of systems. So if you want to actually work from, let's say, a microtiter plate, and your aspiration is to go to a full industrial scale, the best strategy, the most empirical sort of path to follow is what we would call a downscaling system. So here, really what we would look at is, instead of going upwards in incremental steps, which is typically what you see a lot of biotech startups doing, and then they hit these, what I would call like glass ceilings, technical problems that they can't break through. And small sort of insignificant technical details at a lab or a microtiter plate scale are really amplified into potentially big problems when you get to scale. And that sort of obviously links into efficiency and cost competitiveness. So really the best approach, in my opinion, is to use a downscaled system. So what this really means is, firstly, you decide on what's your sort of aspirational scale. This is 100,000 liters, this is where you want to get to. And then you define what are the constraints at that scale. So what are the sort of the physicochemical parameters, and what can you actually achieve at that scale? And then effectively almost make a back plan. So instead of going upwards, you go down, and you design your working backwards from your industrial scale that you've set this sort of framework for. You go back to what a pilot scale system would need to look like to mimic that big scale. And then from the pilot, you can even then mimic a lab scale. And if you're capable and clever enough, you can even make a microtiter plate somewhat mimic a full scale industrial process. But really it's starting sort of, looking where you want to be, and then working out what the journey is, rather than the other way around. And that typically tends to be a lot more successful.
We've been talking mainly about, the fermentation side of things, the actual culturing of the organisms. Like there is a very important downstream processing part, which is also part of the manufacturing process. Again, like any myths around that you would like to pop here?
It's a great call out, and it's one of the things that I think is often most overlooked. Don't forget about downstream processing, DSP. It's a critical piece in the puzzle. In my experience, it's often kind of one of the last things people think about. So when they sort of begin to design strain and process combinations, or start out on a development program, it's typically, it's strain engineering first, it's then upstream processing or the fermentation, and then it's downstream processing almost as an afterthought. But what I would say is, you've really got to look at this sort of more holistically and think of the entire system as one. So as you begin in the first place, and one of the other points you mentioned was modeling. When you first start to design models that are going to simulate your process, you've got to have all of the pieces, all of the cogs of the machine together, because ultimately it doesn't work without all of them. And with regards DSP, what I see a lot is high technology readiness level USP, so fermentation processes and strains, but then coupled with relatively low TRL downstream process system design. And what you can often see is sort of this philosophy of it's almost like, we'll solve that later, we'll kick that problem down the street and we'll deal with that in the scale up. But what you often find is a lot of these issues are extremely more complicated and more expensive to deal with further on down the line. than in the beginning. There are some sort of tricks and things you can do with strain engineering which can solve you a lot of problems further down. So thinking about the downstream processing as well right from the beginning gives you this kind of all-round approach, which gives you overall a much higher success rate and a de-risked sort of strategy that you'll actually get to the scale, your manufacturing scale that's your target.
Yeah, one of my first jobs coming out of my PhD was I was the scale-up guy. I was the guy that basically took it from the lab and kind of like, got it into scale-up. And we were always taught that that if you have a problem in the lab, that you can sort it out in scale-up. And it never, ever happened. They said you could sort of get away with, there were numbers that people would quote that, if you had like 50 mgs per mil in a shake flask, you could do something, in large scale. Like I was never a big fan of that idea. I always thought if it didn't work in the shake flask, it was never going to work in my fermenter. Like, what's your take on that?
I'm completely aligned with that. And again, that's another thing I see a lot. And it links into some of the things I said before, about sort of DSB being an afterthought. But this idea, and I use the phrase like, don't fall into the trap of the sort of the scale up savior syndrome, thinking that sort of we could solve everything with scale up. Once we get to a bigger scale, the equipment will be more efficient. To be honest, for some aspects, for certain things like certain unit operations, a bigger scale is potentially an opportunity to solve some problems. But ultimately, if your process, your strain and your process combination doesn't work at a lab scale, it's highly unlikely that it's going to work anywhere near the efficiency at sort of the bigger scales. So I would always say, look, think of scale up as more of an opportunity to fine tune and to optimize processes. In general, you've got to have it working fairly robust and within, I would say, a line of sight of techno-economic viability, even at lab and pilot scale, before you would even consider really going to sort of demonstration scale and industrial scale. So this comes back a little bit to the modeling. One of the key things really to call out is, when you start off on a process development sort of path, is build as robust a model as you can, incorporate the fermentation, the downstream processing, but build in as much of the techno-economics as you can. So things like, simulating the cost of goods at the scale that you aspire to produce at, because this really gives you your baseline, and then if your theoretical process fails to, let's say, sort of surpass or even meet the break-even point, you've got some big challenges. So it's kind of like a calibration tactic. Models are fantastic, and I wouldn't do any sort of work without strong models, but models are only as good as kind of the assumptions that you make and the data that you really plug into those. So there's no substitute for real world data, which is where things like piloting are such a powerful sort of space.
One final question, like any take on fusions? Like, what's your like, we see a lot of like folks come in, especially at the very early stage, the proteins are fused to tags and that kind of thing. Any take on that?
Fusion, very interesting space. I'm not going to be pretend that I'm the absolute expert on this. I think, to be honest, in my opinion, I think it's an interesting space. I think there's some opportunities with fusion where you could have a particular, let's say, a particular bioproduct that's got a stability issue or an expression or secretion issue, where fusion could be a tool to help overcome some of that. But with every sort of upside, there's a downside. The issue with fusion would be, from sort of my perspective, I would see that as you've effectively fused two things together and created a hybrid of something. So I would be aware of, from a sort of regulatory perspective, on how that's viewed and sort of how that would feed into your timeline. If you've got a commercialization timeline, being aware of this being classified as something, entirely novel, even if it's two things that are bioidentical to something in nature, when you put them together, you've got a composite almost. So I would say, look, opportunities, but also sort of some things to be cautious of at the same time.
Okay. Well, thank you very much, Stuart, for joining us today. like you have to go now, I know. So want to thank you again for sharing your insights. Really appreciate it. Okay, thank you very much, Stuart.
It's been a pleasure. Thank you very much.
Okay, take care.
Bye-bye.
I'm very pleased to welcome Will Canine, who's co-founder and CPO of OpenTrons. Welcome, Will.
Thank you. Good to be here. Always happy to be at an SOSV event.
Yeah, so we're big fans of OpenTrons at IndieBio and SOSV, so more of that later. So just tell us a bit about OpenTrons.
Sure. OpenTrons, our tagline has been, we make robots for biologists. If you look around the world at the 400,000 plus biologists out there, 9 out of 10 of them are pipetting everything by hand and don't have any access to automation. And OpenTrons really started to solve that problem and make lab automation accessible to anyone trying to run life sciences. So, when we looked around the world and thought, how do we automate this 9 out of 10 labs that today don't have any robotics? Well, the first thing is you have to be affordable, because most robotic systems start at like $50,000. Ours starts at $5,000. So that's the first thing that you have to do, is make it so that every lab can afford a robot. But after they buy their robot, then they have to be able to use the robot. So unlike the traditional automation systems, we're geared at the standard bench molecular biologist who's trying to automate their own work, rather than sort of pass it off to an automation tech and let it go to a high throughput core facility, which is sort of the old model. So it's got to be easy to use, affordable, and then open source and customizable is the third thing required to create ubiquitous lab automation. And that's why OpenTrons is open source. And for some customers, that just means that it's really easy to share their protocols back and forth. I do a cool ligation protocol and I want to email it to my friend on the West Coast, and then they can download it and run it themselves. And then for some more hardcore technical users, that means that they're going to integrate their whole confocal microscope into the robot and create a new API that our API talks to, and do a ton of integration and technical work like that. So affordable, easy to use, and open source lab automation is really where we got our start. And the first thing that we did as a company is go to Shenzhen, China, and join HAX, the SOSV program Accelerator. And we still have our own factory in Shenzhen where we manufacture all of our robotic systems. And that has led not only to low costs and good unit economics, but also class leading turnaround time for orders. Right now, we're at about a 3-week turnaround time. And, through COVID, a lot of robotics companies have had more like a 6-month turnaround time. We've been able to scale up our manufacturing. We've doubled it twice in the last 12 months. And we also can come out with a new product really quickly, since when we're designing, we're co-designing with our own factory in the supply chain. And that means that a prototype can pretty instantly go into manufacturing instead of having to be redesigned for manufacturing. So in a nutshell, that's kind of the OpenTrons robotics vision.
Mm-hmm. Like you've very much kind of ridden the SynBio kind of wave, like you're a real kind of like mover in SynBio and helping, like we see it with early stage companies, who come from the SynBio space, and they utilize a lot of automation. So we see a lot of folks that do the use OpenTrons. Can you say anything about the kind of data that they create in terms of the robustness and like what OpenTrons gives to them?
Totally. So when people define SynBio in a lot of different ways, I like sort of the Drew Indy and Tom Knight school that says that SynBio is really using other engineering practices in bioengineering. And what that means in practice is that people really want to write code to run experiments and then analyze data using code as well. So you have this closed loop where code is the connective tissue between the biological processes that you're running. And basically, if you don't have a robot that can run your coded experiments, you don't have a closed loop. And if and so there were people who had these big foundries that cost millions of dollars and have, cloud compute connected to them and all that good stuff. And that's amazing. And you can produce a lot of beautiful things there, but that's not most people. And so if you're going to make it so that a startup in IndieBio can afford a robot so that they can write an experiment in code and then effectively analyze the data in code and then reiterate like that, you gotta, have an affordable, easy to use, open source robot. And so we really filled, I think, a critical niche. Our early adopters are like, diehard favorite, customers at the early stage are almost all of them synthetic biology folks who, they bought our Kickstarter because they've been dying to have a robot that they can write code for in their lab. And now, we launched as an iGEM team, actually, through the GenSpace iGEM team. So our Kickstarter was announced on the floor of iGEM, which is a big synthetic biology collegiate competition. So, we're not a synthetic biology company, obviously. We make robots, but our community started in SynBio, and that really is sort of the early adopter nugget that the rest of our success snowballed from.
Mm-hmm. Where'd you see automation going? Like are experiments just going to get bigger and bigger? Are we just going to go from 96, 384 and kind of like just keep on going? Or do you think there's going to be is there a limit to, like, are we just collecting data? Like, can you say anything? Yeah. I'm thinking a lot about design of experiments and quality by design and that kind of thing.
Right. Well, I mean, I think that there's absolutely no doubt that biology is a big data engineering discipline. You need to create vast data sets in order to draw sufficient conclusions. And that's because biology is just so complex. And without automation, you're just never gonna get there, right? But I also think that since the invention of biotechnology in the 70s with the first discovery of restriction enzymes, we saw Genentech and we've seen big pharma, and the wave after wave is more centralization, more sort of scale through centralization. And I really have started to think that we've started to see some diminishing returns on investments for big centralized biotech. And actually the next important wave is, it's like with computers. It started with servers and then it went to the PC and then it went to the cloud and now it's going to the edge. And I think we're starting to see this explosion, this decentralization in biotechnology now. And that's really the fertile edge of innovation and where we're seeing the most exciting new developments is by empowering folks who are not at MIT and Stanford, not at Pfizer, and who are in communities that need biological solutions. And now for the first time, these smaller teams have the ability to actually build those biological solutions because the tooling is there and there's support networks and an ecosystem that can help them get off the ground. And so, I think that, to go back to answer your question, automation is going to be at the forefront of life sciences expansion, and life science is going to take over everything. And so it's also going, bigger and higher throughput and massive, like it's going there. But it's also, I think, going in this other really more greenfield, unexplored direction of decentralization into the small and mid-sized labs and automate. We were the first lab automation in sub-Saharan Africa, right? Like we're going places that haven't seen this type of technology before. And that's what really excites me, because the use cases these folks have are so important to their livelihood and not that technically challenging. They just need the ability to do it.
Yeah.
And that's really what why I started OpenTrons, is to put the tools of producing these important biological technologies in more people's hands.
Yeah, that's a great point. Like we're talking a lot about distributive manufacturing now and secure supply chains and all that stuff. So I think that's a really good observation that you've made there. Just to sort of talk about, like the past year. While the rest of us have been on the sofa, self-isolating, OpenTrons has been very, very busy. I don't know if you can give us an update or just a quick view of what you've been doing the past year. I know you come to New York.
Yeah, I mean, we've been based in Brooklyn since the beginning, which is, where the 3D printing revolution started with MakerBot. And that's really a lot of our technical stuff is, we have a bunch of people from MakerBot on the team. So New York has been our home for a long time, but that really started meaning something else in the middle of when the pandemic started, and this was really the global epicenter. So just to zoom out a little bit, OpenTrons has been on this track to try and make the perfect robot for all biologists. And that's, a hard thing to do. Like you make the right robot, but then you also have to make the right fulfillment system, and you have to make the right onboarding. You have to make all these things. And OpenTrons has a very different approach. It's all online and self-serve. So the folks get their robot, and instead of paying $10,000 for a technician to come in and install it, they install it themselves. And that's become really important over the pandemic, as you can imagine, doing social distanced robotic installation. So I'm setting this up to say that basically since we raised our Series A and started developing the OT2 and shipping the OT2 in 2018, it's been a march to create the whole product solution that is right for our target market, which is every bench biologist in the world. And really, the end of 2019, the last quarter, is when we really finally knocked down all the final pegs to have that correct solution. And it was just in time for the world to wake up and say, hey, we need a lot more lab robots in the world. And so, it's one of those, every successful startup has some super serendipitous stories in there. And this is really one of ours where we hit that, we built the right features, we've tested them, they work, and we know that we have the right product to go out to the big market. And then COVID hit, and we needed to start shipping 2, 5, 10 times more machines than we ever had before. And the way that, it's, qPCR is how you run a COVID diagnostic, and it's the number one protocol we've automated since the beginning is qPCR prep, but on the academic side, as opposed to on the diagnostic side. And it was March 8th when the FDA issued their emergency use authorization, 2020, that basically said you don't need, it basically got rid of 2/3 of the paperwork required to deploy a COVID diagnostic. And that opened the door for OpenTrons to be used by CLIA certified labs to run COVID diagnostics under emergency use authorization. And so that's really when we snapped into gear, and we started, we were like, okay, what's the best configuration of OpenTrons that you can have to run as many COVID tests as possible? And we came up with a 10 robot, 3 different robotic configurations, 10 total machines, and it can run 2,500 tests a day, basically over 24 hours. And we developed the protocols very, very quickly, and we got them into labs as fast as we possibly could. And we started learning and iterating. And over the first 3 months of the pandemic, we emerged as the best solution for the, I'll call it the tier 2 hospital systems, the not the richest hospital systems, but still, running lots and lots of COVID tests with major requirements. And so, I think we shipped over 300 robots just to hospitals in Spain, a similar number to robots in northern Italy, and then we work very closely with Oxford Nanopore in the UK, where Nanopore runs a lot of the distributed COVID diagnostics in the UK. And then, we're in hospitals and places in the US like Pittsburgh and Baltimore, New Orleans, Oklahoma City, Sunnyvale, California, like places with a lot of population, but not necessarily enough money to buy the Roche Cobas and 200,000 cartridges to use over the course of the pandemic, right? That really, it 5x'd the robots that we were expecting to ship in 2020, just in the first quarter or two of the year. And obviously that tested our manufacturing and our customer service and all that stuff. And seriously, our team has just been heroic in every time zone and taking on challenges that we would not have dreamed of forcing on people without such a crisis and without the potential to have such important impact. And at this point, our machines, excluding the labs that Opentrons ourselves run, which I'm going to get to in a minute, just our robots and other people's labs are running over 3 million COVID tests a month at this point. And 1 million of those in Spain, 1 million of those in the US, and then kind of 1 million throughout the rest of Europe and Latin America and stuff. So, the first half of the pandemic was really just about, all right, let's get these robots in as many labs as we possibly can to help scale up testing. And through that process, we automated a lot of COVID labs. So we learned a lot about the best ways to run a COVID diagnostic. And don't forget that a major factor during this whole time is severe supply chain shortages. So the, tried and true, most gold standard, Roche and QIAGEN and all that stuff, not to be found. You can't get that stuff unless you're willing to pay 5x normal prices, and most people can't do that. And so we ended up establishing completely different supply chains for almost all of the labs that we automated were using a different type of supply chain. And so we were not only able to see the best sort of diagnostic strategies, but the most robust reagent and consumable supply chains that were coming through the United States. And so, that put us in a really good position when the New York City Economic Development Corporation came to us and said, hey, we're trying to build an all new COVID only lab for 10 to 20,000 tests a day by September, this is June of last year. And, we want to bid for you. And so we were able to basically go to one of our customers from NYU, Jeff Buca, a really famous synthetic biologist again, who had basically taken an army of volunteers and converted his genomic foundry into a high throughput COVID testing lab for NYU. And a big important part of that was bolting on a ton of Opentrons into their genomic foundry so that you could convert the sort of clunky samples coming from hospitals into the compact form factor required for their high throughput work cell. And we basically licensed that technology and won the bid from New York City, which meant that today we run the largest COVID testing lab in New York City that's running more than 25,000 tests per day. Both for the public-private hospitals, the 11 public-private hospitals represented by the New York City Economic Development Corporation, but also folks like Northwell and other private healthcare providers. And so that's been, that was basically through 2020 is September through the end of the year, getting that lab really up and running, and to the point that it is now, which is just cruising, running single big COVID testing lab in New York City.
That's an amazing story. Like, congratulations to you and the OpenTrons team. That's a fantastic story. Great journey you've been on, like from those early days when I saw like the early prototype. Like just an amazing journey, amazing story. Yeah.
Thank you.
Okay, well, thank you for joining us today. Really appreciate you coming on and telling us all about OpenTrons and the journey you've done and the amazing contribution that you've made to, especially here in New York, to getting on top of COVID. Really, for myself, I appreciate it. So thanks.
Hey, thanks for having me. And lots more to come from OpenTrons. So look forward to telling you again the next time.
Okay, well, we'll be buying some more OpenTrons, okay?
Excellent. Excellent. All right, thanks.
Okay.